Okay, gastrointestinal physiology. The function of the digestive system is the physical and chemical digestion of food that we eat and the absorption of said food into the bloodstream. Okay, so if you want to trace it, start in the mouth, you go to the pharynx, which is basically the back of the throat, esophagus, stomach, small intestine, large intestine, rectum, anus, and out. Keep this pretty simple. We're going to go over some key terms and then we'll chug through the organs of the GI tract. So mechanical digestion is the physical breakdown of food, primarily in the mouth and the stomach. And then we have chemical digestion, which is the enzymatic cleavage of the chemical bonds that are within the food that we eat. This occurs in the mouth, the stomach, and the small intestine. And after digestion, the nutrients and vitamins and minerals can then be absorbed from the lumen of the GI tract into the circulation. Peristalsis, a word you might be familiar with, it's just the coordinated smooth muscle contractions that propels food along the GI tract. I think of it as like squeezing a tube of toothpaste to get the very last bit out. It's a coordinated contraction, segmental contraction that just pushes food down the GI tract. The term Zymogen is something we'll have to know. So a Zymogen is an inactive precursor of a digestive enzyme. Okay, the reason this is important is because digestive enzymes are meant to chemically modify nutrients. But as you know, our body is made out of proteins and fats and carbohydrates. So if these were active all the time, they would destroy our bodies. Okay, so we have to make them in an inactive form, and we have to activate them only as needed to digest our food. So we make them as zymogens, which contain a catalytic domain and a regulatory domain, and we modify them by typically cleaving off the regulatory domain and allowing them to exert their function. Some examples are pepsinogen and trypsinogen, common suffix there. Okay, so we're just going to go through the organs real quick. The mouth, this is how we eat food. Some important things, antidiuretic hormone and aldosterone stimulate our thirst and make us want to drink water. And glucagon and ghrelin stimulate our hunger and make us want to eat. And leptin and cholecystokinin stimulate satiety or fullness, which prevents us from overeating. Okay, but when we do find it appropriate to eat, we will put food in our mouth and we will chew it. That's mechanical digestion. We will salivate, and enzymes in the saliva, called amylase and lipase, will begin breaking down carbohydrates and fats, respectively. The food will become lubricated into a nice bolus that we can swallow. That bolus will move into the esophagus, which is a muscular tube that propels the bolus via peristalsis into the stomach. One important thing to know is the upper esophagus is skeletal muscle, whereas the lower esophagus is smooth muscle, which should kind of make sense because you can initiate swallowing at will. That's the upper esophagus, but you can't stop the propulsion of that swallowing reflex down to the stomach because the lower esophagus is smooth muscle and therefore mediated by the autonomic nervous system and out of our control. Esophagus goes to the stomach. Stomach is involved in the temporary storage of food as well as continued mechanical and chemical digestion. There are different parts of the stomach. The fundus, right up here, and the body, the main part of it, contain gastric glands with a few different cells that we're going to have to be familiar with. The parietal cells of the stomach secrete HCl, hydrochloric acid, to acidify the lumen of the stomach, and they also secrete intrinsic factor, which will be important for vitamin B12 absorption. Okay, so it's two main things secreted from the parietal cells. Mucus cells will secrete a bicarbonate-rich mucus, and the bicarbonate in this mucus will help to protect the wall of the stomach from the acid that's being secreted into the lumen. And then the chief cells will secrete pepsinogen, which is a zymogen that gets converted to pepsin that begins the digestion of dietary proteins. The lower part of the stomach, the pylorus and the antrum, contain pyloric glands which contain G cells that secrete gastrin. Now, gastrin is a hormone of the stomach, so it travels through the circulation. It is not secreted into the stomach, but it signals the parietal cells to secrete more acid. After the stomach, we move into the small intestine. Small intestine is composed of, in order, the duodenum, the jejunum, and the ileum. I would remember it as Dow Jones Industrial or Dow Jones Index, if anybody ever pays attention to the stock market. So, duodenum, that's an important one. It handles the majority of the chemical digestion of our food. The acidic chyme, which is a term for the modified food that's in the stomach, the chyme entering the duodenum will stimulate the secretion of a couple different things. One of which is secretin, a peptide hormone that will stimulate pancreatic bicarbonate release and inhibit further gastric acid release. This is directly involved in protecting the lining of the duodenum and the rest of the small intestine, protecting it from the acid from the stomach. So we produce more bicarb, we inhibit acid release. Cholecystokinin is also produced upon the introduction of chyme into the small intestine. This will stimulate the bile to be released from the gallbladder. Okay, bile, as you may know, is responsible for helping in the digestion of fats, which we'll talk about in a moment. And it stimulates the release of pancreatic juices, which are bicarbonate rich, help to control the pH, and they contain enzymes that will control the bulk of chemical digestion in the small intestine. In addition to that, there are these things called brush border enzymes. The brush border simply refers to the villi and microvilli that line the surface of the cells that look like a brush, apparently. Okay, so brush border enzymes are released from the wall of the duodenum. Okay, and they include disaccharidases that digest disaccharides. For example, lactase digests lactose. And the importance of this is that carbohydrates must be in their monomer form to be absorbed. So that's glucose, fructose, galactose. They must be monomers so that they can be absorbed. Peptidases from the brush border will digest peptides. For example, aminopeptidase removes the amino terminal amino acid, and dipeptidase can cleave dipeptides. Maybe the most important brush border enzyme is actually enteropeptidase, because enteropeptidase is responsible for the cleavage and activation of an enzyme known as trypsinogen, produced from the pancreas, into its active form, trypsin. And we'll see the role of trypsin in a moment. Okay, so we're in the duodenum, but we're going to take a little detour to the pancreas because it's intricately involved in the function of the duodenum. So, some of those hormones secreted in the small intestine will activate the exocrine pancreas to secrete its pancreatic juices, which contains things like pancreatic amylase. As we know, amylase breaks down starches. Pancreatic peptidases, trypsinogen, chymotrypsinogen, procarboxypeptidases A and B. These are all zymogens. Okay, and back to enteropeptidase from the brush border. The importance of this is it cleaves trypsinogen from the pancreas into trypsin. The active trypsin can then cleave the rest of the zymogens into their active form. Okay, so enteropeptidase is necessary for starting this cascade of converting zymogens into active enzymes that then allow for digestion. And then finally, the pancreas will produce lipase, which helps to break down fats into free fatty acid and glycerol. The gallbladder also is activated to contract based on the hormone cholecystokinin. It's responsible for the storage and concentration of bile. It does not produce bile. Bile is produced by the liver and simply stored in the gallbladder. That's why if you get your gallbladder removed, you still make bile and you can still digest fats. Bile will emulsify fats, which means break down a big droplet into smaller droplets, okay? And breaking a big sphere down into smaller spheres will increase the collective surface area of those spheres, and that allows a greater area for which pancreatic lipase can exert its effect. Okay, so the first part of the small intestine, the duodenum, is intricately linked in function to both the pancreas and the gallbladder, and it's responsible for the majority of chemical digestion. But as we move forward into the jejunum and the ileum, we're going to change the primary function into more absorption. So villi and microvilli in the jejunum and the ileum increase the surface area for absorption. That's their main function. Here is a villus. On the surface of this villus would be smaller projection called microvilli. Importantly, there is a capillary bed running through this villus that allows for the absorption of nutrients into the circulation. And there is something called a lacteal, which is part of the lymphatic system. And you'll see that that is responsible for the absorption of fatty acids into the lymph, which will eventually be delivered back to the circulation. A very quick word on how different things are absorbed. Monosaccharides, like glucose, are typically absorbed along with sodium in something called sodium-linked cotransport. Okay, this is secondary active transport that relies on the gradient that's established by the sodium-potassium ATPase. And one thing I would like to point out here is something called sugar-free Gatorade. Okay, so glucose and sodium are absorbed in a cotransport fashion. So if you have something like Gatorade that is marketed to help you rehydrate yourself with necessary electrolytes like sodium, it can do that. But if they then market it as sugar-free Gatorade to help you avoid calories, which are horrible, then it creates a serious problem because the electrolytes in the sugar-free Gatorade can only be absorbed linked to glucose. So if you're absorbing all these electrolytes without the sugar in the Gatorade, you're not going to get a full effect of all the electrolytes. Okay, so save yourself some money. Don't buy sugar-free Gatorade. Buy the good old sugary Gatorade because it will help you absorb food. Okay, this is also important if you ever go on like a medical trip to a third world country where diarrhea can be a serious problem. When you do something called oral rehydration therapy, you must give electrolytes along with glucose to maximize absorption. Okay, so it's not just my spiel about Gatorade, it's also medically important. That's probably the most important one. As far as the others, amino acids, they can be absorbed as single amino acids or di and tripeptides. Fatty acids, triglycerides will be broken down in the small intestine. They will diffuse across the wall, but then they will reform triglycerides, be packaged into chylomicrons, and absorbed into the lacteal, which will go into the lymphatic system. Moving right along to the large intestine, it is primarily responsible for the reabsorption of water. And another thing that you will have to know, it is colonized by bacteria, and those bacteria make up something called our microbiome. They have various functions, but typically they are good to have. And one of the reasons they're good to have is they help us produce vitamin K. So if you know about vitamin K, you know it's involved in the synthesis of clotting factors, so it helps us avoid profusely bleeding. And it is synthesized by the bacteria that reside within our large intestine. Okay, good. You took my hints. So in a newborn who is born with a sterile gut, they have no bacteria colonizing their gut, they will not be able to produce vitamin K. Now, this can be very dangerous because it leads to something called a hemorrhagic disease of the newborn, okay, which is just profuse bleeding, which can be very dangerous. And what you may notice is that a newborn will almost immediately get a shot of vitamin K in their butt to help them make clotting factors so they don't profusely bleed. Okay, and that's an application of the function of bacteria that reside in the gut to produce vitamin K. Moving on to the liver. Okay, the liver, very important. It's an accessory organ of the GI tract. Okay, and something that we need to point out immediately is the hepatic portal system. So we already talked about what a portal system is. We talked about what it means. It's two capillary beds in series. And in the intestines, there will be capillary beds that are involved in the absorption of water and nutrients. Okay, they will form veins. They will drain into the portal vein. And the portal vein will go directly into the liver, where it branches out into a new capillary bed within the liver. That's the hepatic portal system. The importance of this is that everything we ingest, we want it to be seen by the liver first before anything else sees it. Okay, and we'll talk about that in a moment. But there are other clinically important applications of the portal system. Perfect. So what the portal system also means is that if we have a cancer in our GI tract, say the colon, when it spreads through the blood, the first place it's going to go is the liver before it enters the rest of the systemic circulation. And an application of that is liver metastasis is the most common metastasis in colon cancer because of this effect. Okay, but as I was saying, the hepatic portal system basically allows the liver to see everything that is absorbed from the GI tract. Okay, if there are toxins that we're absorbing, the liver can quickly modify them before they get into the systemic circulation. If there are nutrients, the liver can decide what to do with them before they enter the systemic circulation. This also accounts for a first-pass metabolism effect of oral drugs. So if you take a drug orally, and that drug happens to be metabolized by the liver, it will be absorbed in the GI tract. It will be sent directly to the liver, where it will then be metabolized. Okay? So when you take drugs orally, you get less of an effect than if you directly administer them into the systemic circulation. And that's because the liver is the first thing to see anything that enters the GI tract. Again, a direct application of the hepatic portal system. A little bit more about the liver. It's responsible for the storage of glucose as glycogen, and this helps it maintain normal blood glucose between meals. Okay, so glycogenesis, the formation of glycogen, is stimulated by insulin, and then glycogenolysis and gluconeogenesis is stimulated by glucagon. This is, I think, one of our last questions, so we're almost there, guys. I know it's been a lot today, but this one will probably be very challenging. This will see if you know a little bit about biochemistry, cell biology. It was a tough one, huh? So the answer is glucose 6-phosphatase. So the liver and the skeletal muscle can both store glycogen, okay, and they can both use it, but only the liver can contribute to maintaining blood glucose because it has an enzyme known as glucose 6-phosphatase. If you remember from biochemistry, the phosphorylation of glucose traps it within the cell so it can't leave, okay? And in the processes of gluconeogenesis and glycogenolysis, what you're creating is phosphorylated glucose. So that phosphorylated glucose can be used by the same cell that produces it, or if you have the enzyme glucose 6-phosphatase, you can remove that phosphate so glucose can be exported from the cell into the circulation. Okay, this was a tough question, I admit that. I wanted to challenge you guys. But this is a good example of how the MCAT will integrate multiple disciplines into a single question. So this gives you a little bit of system physiology with some biochemistry. So this is a fair question to ask, but it is very difficult. Okay, almost done. A couple more things to say about the liver. It's got metabolic function. It metabolizes toxins, which we've been talking about a little bit. It converts ammonia to urea in the urea cycle. And it's got a system known as cytochrome P450 that's involved in the metabolism of certain drugs and certain toxins. That's about all you need to know about those things. Okay. It also has a synthetic function in that it synthesizes the majority of the plasma proteins, the most important of which is albumin. So albumin is essential for binding and transport of hydrophobic substances. And it's also essential for the maintenance of plasma colloid oncotic pressure. So I alluded to the Starling forces before, but I'm going to allude to them again. And one of the roles of this is preventing edema or preventing fluid accumulation in the extracellular space, because the protein within the capillaries, albumin produced by the liver, can help to maintain fluid in the capillaries, because like other solutes, it's osmotically active. Okay, so that's an application of osmosis as it relates to the synthetic properties of the liver. And then it's also involved in the synthesis of cholesterol and lipoproteins. It's got a lot of roles. It's got a lot more than that, but those are the main ones you need to know.